So real pleasure to introduce our first speaker, so Dek Woolfson from the School of Chemistry, Bristol. Dek has been a protein designer for his entire career. In fact, yesterday, Dek was awarded a fellowship of the Royal Society. So big congratulations to Dek. And just to say that with the power of AI and what it's done for protein design, even Dek, who is a rational protein designer, has started to use AI in his research. It was also suggested, by the way, that this is your inaugural talk. Daryl suggested that you should consider this your inaugural talk for for your FRS. Anyway, can I invite you to? Yes.
Shall I get going? So most of you know me, some of you don't. Those that do know me know that I get carried away, so I'm going to get the timer going for 18 minutes, wasn't it?
10.
I'm going to set it for 8. Okay. And then I'll just make sure I can wrap up to that. So we've got a pointer. Cheap seats, can you hear me okay?
Yeah, really sorry.
I'm going to stand over here. Oh shit, I don't know. Really cramps my style. Okay, I will try. Yeah, it's okay, I'll try. So a few things just to get started. It's a real honour to be kicking this off and to have the luxury of so much time to talk. And I'll try not to abuse that and be on time. Also, I think it's a really exciting time for all the reasons that Ross has just said. And I think it's also a really exciting time for another area that this impinges on, that's synthetic biology. And I can't give too much away, but I know there's now a new Jedi Council at the university in synthetic biology, and I'm really excited to see what happens there in this sphere. The fun thing I want to say, and this is as just as an introduction, is that despite all these advances, I hope you're with me in that we don't understand anything, right, about proteins. And AI just gives us those tools to get into places that we really don't understand. And I'm hoping that we'll have something like an explainable AI in years to come that will actually combine the kind of stuff I love as a physical scientist, as a chemist and a physical biochemist, and the power of AI. Because I kind of think without that we're a bit lost, to be absolutely honest. I think we need this relationship between chemical sequence structure and ultimately function. My backdrop here it looks pretty impressive, right? These are all X-ray crystal structures of de novo designed proteins. None of them are built with AI. They're all built rationally or they're built computationally. All right, that's just exploded. We now have hundreds of protein structures because of all of these methods plus AI. So I thought I'd better just set the scene. Protein folding is the forward reaction, taking a protein sequence, or a linear stretch of amino acids, folding up into a beautiful structure. Protein engineering is the idea that if you change one or a small number of amino acids in that natural sequence, you're doing a protein engineering experiment. That's part of the forward protein folding reaction. The reverse of that, where you take a protein structure or a protein function and you want to generate or design a protein sequence that will fold to that, is the protein design problem or the inverse protein folding problem. Is everyone kind of with me? And of course, the states of the art of these things, AlphaFold and others going forward. The reverse is now, as Ross introduced, generative AI. And this won the Nobel Prize. These two things won the Nobel Prize. So David Bacon, protein design, and Demis and John for AlphaFold. Why do I do it? I've already given this away. I want to understand sequence structure relationships, so I can go in there with like really scalpel-like precision and make changes to de novo proteins to make them functional. But what other people are doing, of course, Ross does beautiful work in this. This is trying to make a catalytic protein. This isn't Ross's work. This is actually a Baker design from computational design with Berta Herka. It's 10% of all the enzymes have this structure. If you don't know it, it's called the Tim barrel. 10% of all the enzymes have this structure. That means it's a privileged protein structure. My understanding is from Don Hilvert, who's probably one of the best protein engineers on the planet, he can't evolve that into a functional enzyme. So even though we can build this as de novo designers, Ross might correct me on this, people haven't really succeeded in changing this particular scaffold, a privileged scaffold, into a catalyst, right? That's worrisome. This is something we built. It's a switch between two states, an open and closed state of a helical bundle. You can use those things to sense metabolites, things for biomedical sensing, for example. This is a protein in colour from Bruno Correa's group, where he can make a picomolar, really tight binder, better affinities than antibodies, to a protein from a virus that he wants to neutralise, right? So that's the hope. The way people do this, sort of been introduced minimally, that's just using very simple chemical principles, separate hydrophobic and polar residues. Rationally, that's embellishing those with our bioinformatic and chemical understanding of proteins to put those into designs computationally, so we can look at all manner of sequences that might fold up into the protein that we want. And now with AI. Just to put this into perspective, this is how we do things. So we use chemistry-based or physics-based rules to build helical assemblies. So we can instruct small polypeptide chains to fold up to find 2 copies of themselves, 3 copies of themselves, and so on, all manner of combinations of those things to produce discrete protein objects. And that's just using pen and paper in biro design, if you like. We can do that because we have lots of examples of this in the literature or in nature. Where we don't have so many examples, these are called alpha helical barrels. We just discovered them by chance, by messing around, right? And we found some proteins with holes in them. There are very, very few examples of these in the natural sequences and structures to learn from. So we had to build our own physics-based computational methods to build these things. And then what Ross alluded to again is that we can use this understanding that we built from rational or computational things, and we can now use AI to join those things up. Now, all of those things on the top 2 lines, they are small proteins called peptides that we build chemically with a peptide synthesizer. These things on the bottom are real proteins. We make a synthetic gene and we express that in E. coli, and they fold as we predicted them to fold, right? But we use the things on the top as seeds for our computational design, and this is aided and abetted by AI. That stuff on the top took my group, really talented people, some of them sitting here actually, 15 years to do, right? Then AI came along and we were able to take those rules that we learned and make these proteins in less than 5 constructs. We make no more than 5 genes. And we always get a crystal structure, and they look like what we predicted in the computer. What are we doing with these things now? Ross does similar things, other people do similar things, Paul does. We've built very, very simple physics-based methods, not AI-based methods, although we use AI in the pipeline, to build holes in proteins that will bind small molecules. And we've bound natural flavin molecule. This is a one-shot design. One design, one gene, it binds our target flavin with micromolar affinity. I will pass over this because I guess that Jess might talk about this. But essentially, we can also build proteins that will take things into cells and hijack processes in cells. In this case, we're hijacking a molecular motor, and we can get the design protein to carry things through the cell on the molecular motor. We can also build materials inside cells. So these are liquid-liquid phase separated systems or biomolecular condensates, very trendy, bit weird in biology. But we can build these de novo, and they're collections of de novo proteins to which we can sponge in de novo chemical functions. That's 8 minutes, so I've still got 2. Is that okay? Yeah, great. But I'm pretty much finished there, right? So here are some questions and some conclusions that we can maybe talk about later on. We can maybe put this up later. Just my indulgence as being the keynote to leave you with those. But what I wanted to tell you about in 1 minute is that I've just been asked to start a new Centre for Protein Design in Copenhagen. And this isn't going to be introspective. This is going to be broad and open. And Bristol is going to be one of the spokes for this. So we're going to have 3 themes. I've already outlined those. How can we program de novo design fully? How can we do protein design in the context of a cell or a reaction? And then how can we use promoting design in health and sustainability. As I say, we're going to reach out. We're already reaching out across Europe. Bristol should be on there. Oh, yeah, it's on there in a minute. We're reaching out to UCSF. That's going to be one of our spokes. And Bristol's going to be one of our spokes. So there are going to be real opportunities for people to dip into this centre from Bristol. And that's about all I want to say. And we're going to have mechanisms for collaboration. But that's about it. Thank you.